The overall goal of this experiment is to establish a cell-free assay quantifying the neutralizing capacity of GM-CSF autoantibody. We start by obtaining transgenic silkworms bearing the human SGMR alpha gene to produce recombinant SGMR alpha and proceed to label the recombinant human GM-CSF through biotinylation. Next, we evaluate the correlation between the cell-free system and a regular bioassay with a study of inhibition of GM-CSF binding to SGMR alpha by GM-CSF autoantibodies. Results obtained demonstrate a significant correlation between growth inhibition of the bioassay and binding inhibition of the cell-free system by serum IgG fractions and purified GM-CSF autoantibodies.
The main advantage of this technique over existing methods, like TF1 bioassay, is that this system renders a high-throughput analysis with good reproducibility to measure neutralizing capacity of GM-CSF autoantibody. Since GM-CSF autoantibody causes autoimmune pulmonary alveolar proteinosis, the implications of this technique extend toward diagnosis. It can also be applied to competitive binding assays using recombinant cytokine receptors, such as receptors to VEGF and TNF-alpha.
Demonstrating the procedure will be presented by Shinya Urano, a PhD student from my laboratory. He first developed this cell-free system.
This video shows the silkworm used for the production of recombinant SGMR alpha. The first step is to amplify CD.